بسم الله الرحمن الرحيم، والصلاة والسلام على رسولنا الكريم محمد وعلى آله وصحبه أجمعين. بنبلش اليوم المحاضرة الأخيرة في البايوكيمستري ثيوري اللي هي الفيتامينز، ورايحين في هذه المحاضرة ندرس 3 فيتامينات رئيسية وهي B9, B12 and فيتامين C. أوكي. Vitamins. Vitamins are organic molecules, so they are organic. Our body can't synthesize them in enough quantity. Our body can synthesize some of them in small quantity and can't synthesize others at all. In general, we have to get the vitamins, nearly all vitamins, from our food. So vitamins are organic molecules that our body can't synthesize in enough quantity. This is why we have to get them in our food. According to their solubility, vitamins are divided into two groups. Fat-soluble vitamins, which include vitamin A, D, E, and K. And the other is water-soluble vitamins, which include vitamin B and C. So in today's lecture, we will study the water-soluble vitamins, some vitamin of the B, and we will study C also. The water-soluble vitamins, because they are water-soluble vitamins, our body can't store them for long time, except vitamin B12. So in general, the water-soluble vitamins, our body can't store them for a long time, except vitamin B12. While fat-soluble vitamins, our body can store them for longer time. Now, we will start with folate, which is also known as vitamin B9. So we can call it folate or vitamin B9, and previously it was known also as folacin. As you heard before also, it is also known as folic acid. What is the difference between folate or vitamin B9 and folic acid? The natural vitamin B9 or natural folate, we call it folate or vitamin B9. This is the natural one in nature. While the synthesized one, the one that the pharmaceutical companies made, we don't call it folate or vitamin B9, we call it folic acid. So the natural one is folate or vitamin B9, while the synthesized one, we call it folic acid. Folate are synthesized by bacteria and plants. So our body can't synthesize the folate or vitamin B9. And in plants, it is mainly found in dark green leaves, in beans, peas. Also, it is found in the fruits, orange, lemon, strawberry, melons, and so on. So our body can't synthesize folate and can't store it also for a long time. This is why we have to get it regularly in our food. Natural folate, as we will see, natural folate or natural B9, vitamin B9, are always naturally conjugated with glutamic acid or glutamate. This is why we call it polyglutamyl chain. So B9, vitamin B9 is always attached with what? With the amino acid glutamic acid or glutamyl or glutamate. So it has a number of glutamic acid attached to the vitamin B9. This is why we call it poly. Poly means many. Glutamic acid, the amino acid. But see here, So vitamin B9 is always naturally attached to polyglutamine, but once in the brush border in the small intestine, once in the small intestine, the enzyme folate conjugase cleave the polyglutamate chain from B9 and leaves only one glutamate attached to B9. This is why it is known as monoglutamate, folate monoglutamate. So naturally, the folate is always attached with polyglutamate. In the small intestine, the enzyme folate conjugase cleaves the polyglutamate and leaves only one glutamic acid only attached to folate. This is why now in the small intestine, it is found as folate monoglutamate. The folate that enter to our body and our blood is in the form of 5-methyl tetrahydrofolate monoglutamate. 5-methyl tetrahydrofolate monoglutamate, as we will see. Folate chemical structure, as we will see in the next slide. Folate is made up of pyridine ring. So folate is made up of pyridine ring. Pyridine ring is made up of two fused rings. They are pyrimidine and pyrazine ring. So pyridine ring, as we will see, let us go to the second. This is the structure of the folate. So the folate is made up of one pyridine ring. This is the pyridine ring. It is made up of pyrimidine ring and pyrazine ring. 6-membrane ring joined together. 1 and 2. Together, what do we call them? Pyridine ring. They are fused with each other. The second is PAPA. PAPA is the short form of para, as you can see from the structure, para aminobenzenic acid. Para aminobenzenic acid. Amino because of the amino group. Benzo because of the benzene ring. Acid because of the COH. So para aminobenzenic acid, PAPA. Para aminobenzenic acid. And the last, as we said, is glutamate or glutamic acid. As we said, and as you can see here, N. Naturally, it is polyglutamate. But once in the small intestine, the poly is cleaved. And what remains? That enzyme keeps only one glutamate attached to PAPA and pyridine ring. This is the structure of the folate. So it is made up of pyridine ring. Pyridine ring is made up of two fused rings. They are pyrimidine and pyrazine ring. And PAPA, para-aminobenzenic acid. Para, as you can see, 1 and 4. Amino group, the NH, benzene, and the acid. And the last is glutamate. In the nature, as we said, it is poly, but in the small intestine, it is mono, only one glutamate is there. Folate function. Folate plays a very, very, very important role in our body, specifically in the synthesis, repair, and methylation of DNA. See how important the folate is. It plays a core role in the synthesis, repair, and methylation of DNA. And thus, it plays a very important role in cell division. As we will see. Also, it is a cofactor in many biological reactions, as we will see also. And also in the production of healthy red blood cells. And their shortness causes some type of anemia, as we will see. So this is the folate function. So folate or vitamin B9 play a very important role in our body. In the synthesis, repair and methylation of DNA, in cell division, production of healthy red blood cells, and a cofactor in many biological reactions in our body. Folate activation. Folate in its form, the one that we study, is not active. So folate in its structure, the one that we studied, is not active. How the folate is activated? By donating to them 4 hydrogen. See? 1, 2, 3, 4. To which atoms? 5, 6, 7, and 8. So the activation of the folate occurs through what? Through donating, reducing them, through donating 1, 2, 3, and 4 hydrogens. This is why now the folate is known as tetrahydrofolate. Tetra means 4. Hydro, the hydrogen, and folate. So 4, what is the meaning of tetrahydrofolate? It means 4 hydrogens are bonded now with the folate. To which atoms? Atoms number 5, 6, 7, and 8. What is the name of the enzyme that place these or donate these 4 hydrogen? We call it dihydrofolate reductase. Dihydrofolate reductase here. This process or this reaction occurs in 2 steps. The dihydroreductase first give the 2 hydrogen to folate, thus converting the folate into dihydrofolate. Now it has 2 hydrogen. Then the same enzyme in the second step donate the second hydrogen atoms, then converting the dihydrofolate into tetrahydrofolate. And this reaction needs 2 NADPH, 1 NADPH in every step. So this is the tetrahydrofolate. It is the same as folate, but with 4 hydrogens on atoms 5, 6, 7, 8, and this is why it is known as tetra, 4 hydrogen bonded to folate, and shortly it is known as FH4. FH4 is the short name of tetrahydrofolate. Now, the tetrahydrofolate or FH4 play a very important role. As we said about the function of the tetrahydrofolate, we said that the tetrahydrofolate or vitamin B9, the active form of B9, vitamin B9, play a very important role in the synthesis, repair, and methylation of DNA, and also in the cell division, and also play a cofactor in many biological reactions and in the production of healthy red blood cells. Now, see here, and this is very important. Most of the activity of the FH4 comes through transporting the 1 carbon atom. So FH4 is very important. It plays a very important functions in our body, the one that we studied. Through what? Through transporting. This is the mechanism of its function. So FH4 do its function through transporting the 1 carbon atom, a group. So it is 1 atom of carbon through a group of atoms. So, but this group has only 1 carbon. The FH4, the active form of B9, vitamin B9, gets the 1 carbon atom from serine or glycine or histidine or formaldehyde or formate. Then this 1 carbon atom is attached to where? To N5 or N10 in the NFH4. So FH4 take one carbon group from one of these molecules, and this one carbon group is attached to where? It is attached to FH4 atom N5 or N10, or a bridge between them. They share with each other. So FH4, the main rule of FH4, the act of B9, is transporting the one carbon group. One carbon group. This one carbon group is attached where in the FH4? It is attached to N5 or N10 or between them. Examples. See, and these examples are very important. The FH4 transfer one carbon group in the form of methyl group. See, the one carbon group in the form of methyl group. Methyl has one carbon atom only. One carbon group to deoxyuridine monophosphate dump to form deoxythymidine monophosphate, DTMB. This is very, very, very important. As you studied in the cell biology and in your school, uracil is a nitrogen base found in RNA, while thymine is a nitrogen base found in DNA. This is why FH4 donate a methyl group to deoxyuridine monophosphate, the nitrogen base in RNA, thus converting it to deoxythymidine monophosphate, a nitrogen base in the DNA. This is why this step is very, very important. It's very important for the synthesis of DNA, for the synthesis of DNA, thymidine, deoxythymidine monophosphate, as we will study also, inshallah, in the second semester in the molecular biology. Second, also FH4 transfer one carbon group in the form of formal group to purine bases. Purine is adenine and guanine, while pyrimidine is thymine and cytosine. So FH4 also transfer one carbon group in the form of what? Formal group, COHR, to where? To adenine and guanine to produce or to donate or to synthesize or to build which carbon atom in adenine and guanine? C2 and C8. So C2 and C8 in adenine and guanine, the one that is found in the DNA and RNA also, from where it comes, these atoms? It comes from the FH4 that transport them in the form of formal group. This is very important. This is why the FH4 is required in the cell division and also for the synthesis and buildup of the nitrogen bases in the DNA and RNA. This is very important. Also, the FH4 transfer one carbon group to glycine to form serine. So it converts glycine to serine by donating one carbon atom to glycine. This is the function of the FH4 and how it functions. This is the mechanism of the function of FH4. The next slide is about number 1 and number 2. To clarify this slide is very important. See here. And pay attention with me here. See here. Deoxyuridine monophosphate dump, the one that we studied just now, dump, this is the dump, is converted to deoxythymidine monophosphate, as we said, by via the FH4. So FH4, as you can see, donate one carbon group in the form of methylene to dump, thus converting it into DTMB. And DTMB is a nucleotide in the DNA, thymidine. See here, as we said, FH4, it transfers through which carbon atom? Which atoms? And 5 and 10, as we said. It transports one carbon group in the form of methylene and donate this one carbon atom in the form of methylene to dump, thus converting dump into DTMB. And the FH4 is converted to FH2 only. Then, as we said, dihydroreductase in the liver and other cells also will convert FH2 into FH4, and it needs one NADPH, as we said, in each step. Thus, dihydrofolate reductase, as we said, will convert FH2 by donating 2 hydrogens atoms to FH2, thus converting it into FH4. Then FH4 take one carbon group in the form of methylene from serine, which is converted to glycine. So FH4 take one carbon group from serine to be converted to FH4 methylene, and so on. See here, which is very important. There is a drug known as 5-fluorouracil. This drug blocks and prevents FH4 from donating its one carbon group to dump. Thus, dump is not converted to DTMB. This drug is a chemotherapy in cancer. See how important the thing is. If we prevent the conversion of dump to DTMB, thymidine monophosphate, deoxythymidine monophosphate, is required for the synthesis of DNA. If there is no nucleotide thymidine, there will be no synthesis of the DNA. This is why the cancer will be blocked. Another drug, methotrexate. Methotrexate prevent dihydrofolate reductase from converting FH2 into FH4. Thus, FH4 can't donate the one carbon group to dump, thus is not converted to DTMB. This is the chemotherapy drugs, 5-fluorouracil, methotrexate. They are very important. They are very important. And as you know, cancer is uncontrolled cell division. If we block the cell division, we cured the cancer. Thus, if we prevent FH4 from donating its one carbon group to dump, it will not be converted to DTMB. Thus, the nucleotide DTMB will not be available for the synthesis of DNA. If also we prevent the conversion of FH2 to FH4 by methotrexate, then we block the synthesis of DNA. Now, folate deficiency. What is the causes? What causes the folate deficiency? Maybe first, inadequate folate in our food. Inadequate folate in our food. Maybe we are not taking enough food that has vitamin B9. Second, the folate is absorbed in jejunum. Jejunum is one part of the small intestine. Thus, some diseases that occur for jejunum may prevent or block the absorption of folate. So, diseases to jejunum. Why jejunum? Because the folate is mainly absorbed in the jejunum, which is a part of the small intestine. Third, some drugs may inhibit the absorption or conversion of folate to its active form. 4. Congenital deficiencies. What is the congenital? It is the genetic diseases. Genetic diseases that causes the deficiency of some enzyme that is required for folate metabolism or folate activation. 5. Vitamin B9, and pay attention to point 5. Vitamin B12. What is the relationship between vitamin B12 and B9? See, this is the vitamin B12. So the deficiency in vitamin B12 will cause a deficiency in vitamin B9 or the folate or FH4. See, so there is a relationship between vitamin B12 and vitamin B9. Vitamin B12 is required by an enzyme known as methionine synthase. To what? To remove the methyl group attached to where? To N5 in the FH4. As we said, FH4 transport the one carbon group, and this one carbon group is attached to which carbon atoms in the FH4? It is attached to N5 or N10 or between them. Methionine synthase is specific to remove methyl group from N5 in the on FH4. And this enzyme needs B12 for its activity. So methionine synthase require what? Vitamin B12 for its activity. This is why if there is deficiency in vitamin B12, methionine synthase can't remove the methyl group attached to N5 in the FH4. So this will cause that FH4 is always attached to it, to N5 of FH4, a methyl group. So it can't donate the methyl group. It can't function. This thing is known as trapped. Trapped يعني في كل الأحيان ال FH4 مرتبط بال N5 ما بيقدر يعملها donation. So a functional folate deficiency result those carbon can't be removed from folate. So the methyl group attached to N5 in the FH4 can't be removed. This is why FH4 can't function properly due to the absence of B12. Much more folate than the normal person needs because she is pregnant. It needs more for her infant or the fetus. Complication. What happened of the folate? Complication of folate deficiency. The untreated folate deficiency also known as macrocytic anemia. Megaloblastic anemia. It is characterized by reduction in the number of mature healthy red blood cells. Red blood cells, also known as erythrocytes, RBCs or red blood cells, all the same. So folate, as we said at the start of this lecture, we said that the folate is very important for the maturation of red blood cells. for the maturation of the red blood cells. And also, this condition is caused by impaired DNA synthesis due to deficiency in the folate. Folate is very important for the synthesis of the DNA. For the synthesis of the DNA. The folate deficiency will cause reduction in the mature healthy red blood cells and the production of unmature red blood cells, which are characterized by large size and that can't do their work properly. So in megaloblastic anemia, the red blood cells are large in size and mature and can't do their job properly in transporting oxygen. Second complication of folate deficiency is neural tube defect. The neural tube in the fetus في الجنين في رحم الأم في بدايته. The neural tube is the one that forms the brain and spine. So neural tube in the fetus will develop to form the brain and the spine, which is caused, so this case, neural tube defect is caused by deficiency in the folate during pregnancy. This is why, as you can, if you heard, the pregnant women are advised to take folic acid during their pregnancy to prevent the neural tube defect. Third, congenital impairment, dementia and depression. What is the congenital impairment? Any person that has trouble remembering or learning new things. The severe condition is dementia and also the depression. We finished with the B9. Now we will start with the second vitamin in our lecture, which is vitamin B9, which is known as also cobalamin. Why do we call vitamin B12 cobalamin? Because vitamin B12 has the mineral known as cobalt. This is why we call it cobalamin. So which mineral is present in vitamin B12? Cobalt. This is why we call it cobalamin. See, vitamin B12 is produced only by bacteria. See? So it is not produced by plants. It is not produced by animals. The only one or creature that produces vitamin B12 is bacteria. What is the source of vitamin B12? Which food is rich in B12? In meat, the meat, liver, eggs, dairy product, fish, poultry, and seafood. But the meat and the liver and the eggs and dairy product, from where the B12 comes from? It comes from bacteria. So the meat has B12, but it comes from bacteria. The liver also has B12, but from bacteria. So the only source that produces B12 is bacteria. Now, vitamin B12 structure. Vitamin B12 structure. Let's go to this is the structure. The B12 structure is complicated structure, is a complex, as you can see. As you can see, this is the structure of B12, and it is one of the most complicated structures, as you can see. But see here, the B12 is made up of choline ring. This is the choline ring. Choline ring. It is made up of, and pay attention where the rings are. It is made up of 1, 2, 3, and 4 pyrrole rings attached with each other. This is how they are attached, methylene bridge, and here directly. So this is the rings, 1, 1, 2, 3, and 4. 4 beryl ring. And in the center there is the cobalt. In the center there is the cobalt. As we will study, inshallah, in the third year in the heme. Heme is the one, the protein that is found in the center of hemoglobin and myoglobin. Instead of cobalt, there is iron. Iron. So 1, 2, 3, 4 beryl ring, and in the center there is cobalt. Cobalt makes 6 coordinated covalent bond. How many? 6 coordinated covalent bond. 4 of these bonds to the nitrogen of the beryl ring. 1, 2, 3, 4. The 5th bond with the structure, this structure, which is 5,6-dimethylbenzyl imidazole. And the 6th with X. X means different molecules, as we can see. So B12, this is the main structure for all B12, but X is different from one B12 to another. So B12 is not one form, although this is the basic structure for all B12. But what is the difference between one B12 and another is what it is attached to in the form of X here. Let's go back here. So, as we studied, vitamin B12 is made up of corin ring, which is 4 beryl ring. And in the center there is cobalt. Cobalt makes how many covalent bonds? 6 coordinated bonds. 4, as we studied, 4 to the nitrogen of the corin rings with the beryl ring. Then a 5th with the 5,6-dimethylbenzyl imidazole. And the 6th with the X. X, see here. X could be 5-deoxyadenosine. Then the B12, if 5-deoxyadenosine is there instead of X, then the B12 is known as 5-deoxyadenylyl cobalamin. If methyl group is in the X, then the B12 is known as methylcobalamin. If OH group is in the X, then the B12 is known as hydroxycobalamin. If cyanide in the X, then that B12 is known as cyanocobalamin. So this is the 1, 2, 3 form different of B12. But the 5-deoxyadenosine and methylcobalamin are the metabolic active form of B12. So the active form of B12 are 5-deoxyadenylyl cobalamin and methylcobalamin. So this is the structure of B12. Now, absorption of B12. Absorption of B12 in the small intestine. See here, the B12 in our food, B12 could be free, not bonded with a protein, or could be found bonded to protein. So B12 in our food, once you had your food, the B12 could be free, not bonded with a protein, or it could be bonded with a protein. See here, the free B12 will bind directly to transcobalamin 1. Transcobalamin 1 is also known as R binder and haptocorins. So we can call it transcobalamin 1. We can also call it R binder, and we can also call it haptocorins. 3 names. So the free B12 will bind once it's absorbed, it will bind or in our gastric food. Once we ingest the food, the free B12 will bind to transcopolamine 1, which is secreted by our salivary gland and gastric mucosal cell in the stomach. So free B12 will bind directly to transcopolamine 1, which is also known as R binder, as we said. What about the B12 that is bonded with the protein? The B12 bonded to protein. The protein that is bonded with B12 will be digested by proteases. What is the proteases? Proteases are a group of enzymes that digest proteins. So all enzymes that digest the proteins, we call them proteases as a group. So proteases will digest the protein bonded to B12, thus releasing a free B12. Once the B12 is free, it will be again, it will bind to transcopolamine 1. Thus, now all B12 now in the small intestine, all B12 are bonded with transcopolamine 1. Then, what happened? The pancreatic proteases in the small intestine, pancreatic proteases in the small intestine digest transcopolamine 1, destroy it. Digest transcopolamine 1. Then B12 is not bonded with anything. Now it will bind to intrinsic factor. Intrinsic factor, which is a glycoprotein, carbohydrate with a protein. So intrinsic factor synthesized by the parietal cells of the stomach. So in the small intestine, B12 will be bonded with the transcopolamine 1. In the small intestine, transcopolamine 1 will be destroyed, will be digested by proteases, enzymes, proteases, pancreatic proteases. Then the free B12, now again free, will bind to intrinsic factor. This intrinsic factor binds to B12. What is the function of intrinsic factor? For the absorption of B12 in the ileum. Without intrinsic factor, our small intestine can't absorb the B12 vitamin. So the intrinsic factor, as we will see also some diseases, without intrinsic factor, our body can't absorb the vitamin B12. So the intrinsic factor binds to B12, which is a glycoprotein, and this intrinsic factor binds to a receptor C. So the intrinsic factor, which is a glycoprotein, binds to its receptor in the ileum. Ileum is a part of the small intestine, one part of our small intestine. So intrinsic factor, which is bonded to B12, or together, they bind to their receptors in the ileum, and their absorption occurs there. This is why without intrinsic factor, B12 can't bind to its receptor in the ileum, and there will be deficiency if there is no intrinsic factor. Then, inside the ileum cells, inside the ileum epithelial absorptive cells, B12 will bind to another transcopolamine 2 here, 2. So the absorbed vitamin B12 will bind to transcopolamine 2, which will be transported to the liver. The liver will take 50% of the absorbed vitamin B12, and the rest will be transported to the cells where it is needed. So 50% of the absorbed vitamin B12 will be stored in the liver. This is why, see, which is a very important information. This is why the amount of vitamin B12 stored in the liver is enough for our body for 3 to 6 years. See how huge the B12 in the liver, how huge the storage is. So even if there is deficiency in your food, the liver storage is enough for our body for 3 to 6 years. Now, reaction stimulated by vitamin B12. B12 is involved in main two reactions. So, where the B12 is needed in the reaction. Methylation of homocysteine to methionine. This is very important for DNA synthesis and for also melan synthesis in the neurons, also for neurotransmitters, as you will see also in the future, inshallah. Neurotransmitters that transport the action potential between the neurons and also for brain metabolism and growth. So B12 is very important for methylation of homocysteine to methionine. Homocysteine is an amino acid. See why homocysteine is important here as indicator in our body. Because vitamin B12, B6, and folate, the B9, break down homocysteine to create many different chemicals in our body. This is why if there is high level of homocysteine, it means there is a deficiency in one of these vitamins, or maybe all of them. So homocysteine is very important indicator. Why? Because vitamin B12, B6, and folate, they all together break down homocysteine to synthesize different chemicals in our body. This is what keeps homocysteine in normal level in our body. Thus, if there is high level of homocysteine in our body, it means there is a deficiency of one or more than one of these vitamins. The second reaction, conversion of L-methylmalonyl coenzyme A to succinyl coenzyme A. This reaction is very important for the production of energy from fat and protein. So B12 convert L-methylmalonyl coenzyme A to succinyl coenzyme A, which is very important, as we said, in the production of energy from fats and protein. This conversion is catalyzed by what enzyme? Methylmalonyl coenzyme mutase. So where the B12 is, this enzyme to function needs vitamin B12. So this enzyme, methylmalonyl coenzyme mutase, require B12 to convert L-methylmalonyl coenzyme A to succinyl coenzyme A. If there is deficiency in vitamin B12, this reaction will be ceased, will stop. This is where the B12 in the reactions in our body is needed. Causes of vitamin B12 deficiency. What causes vitamin B12 deficiency? Pernicious anemia. Pernicious anemia is an autoimmune disease. If you remember, in a few minutes ago, we said that the intrinsic factor produced by our gastric bacterial cells is very important for the absorption of vitamin B12. It binds to the receptors. In the small intestine, and this is what facilitates its absorption. Without intrinsic factor, our body can't absorb B12. This is normal case. Now, what happens in the pernicious anemia? In pernicious anemia, it is an autoimmune disease. What is the autoimmune? The immune system in our body fight the foreign pathogens, bacteria, viruses, molds, yeast and whatever. So it fight foreign pathogens. This is the normal one. Sometimes the immune system fight the person's own organs. And this is, we call it autoimmune disease. In the pernicious anemia, it is an autoimmune disease. In there, the immune system in those patients, the immune system destroy the peritoneal cells in the gastric cell, or in the gastric body, in our body. So it fight and destroy the peritoneal cells. Thus, these cells won't produce the intrinsic factor. This is why the body of these patients can't absorb vitamin B12. Second causes of vitamin B12 deficiency, surgery to the gastrointestinal tract. Third, prolonged taking of some medication. So some drugs may cause also some vitamin B12 deficiency. Diet deficiency. Maybe those persons, they don't eat the food that is rich in B12, as we said. Symptoms of vitamin B12 deficiency: tingling and numbness in the extremities. extremities يعني الأطراف. يعني بيكون في هناك tingling و numbness in the extremities. يعني بيكون بالأطراف في يعني numbness, tingling. وفي عندنا كمان nerve damage and memory loss. So this is the most important thing, is tingling and numbness in the extremities, الأطراف تصير عندنا tingling and memory loss. This is the most two important symptoms of vitamin B12 deficiency. We finished with the vitamin B12. The last things in today's lecture is vitamin C, L-ascorbic acid. The active form of vitamin C is ascorbate. So ascorbate is the active form of vitamin C. Humans are unable to synthesize vitamin C. And as we said before, because it is water-soluble vitamin, our body can't store them for a long time. Vitamin C, as you know, is found in the citrus fruit, berries, potato, tomato, peppers, and others. The main function of ascorbate or vitamin C, it is a reducing agent. It donates electrons in many different reactions. It reducing also agents. See the last point. It regenerates the reduced form of vitamin E, as we will see now. Also, biosynthesis of collagen. Biosynthesis of collagen and absorption of dietary iron from the intestine. See here, as we said, the active form of vitamin C, we call it L-ascorbate. What is the main function of ascorbic acid? It is a reducing agent, as we said. What is the reducing agent? As I told you before, it donates electrons. So L-ascorbate, the active form of vitamin C, donate 2 electrons. It donates in 2 steps. It donates the first electron. Thus, L-ascorbate is converted to ascorbyl radical, then donate the second electron. Thus, it is converted to dihydroascorbic acid. So, the reduced form, the active form of vitamin C, we call it L-ascorbate. The oxidized form of ascorbic acid, we call it dihydroascorbic acid. And ascorbyl radical is intermediate molecule. Intermediate molecule. So L-ascorbate or ascorbic acid or vitamin C, what is the main function? A reducing agent. Reducing agent. It donates electrons in 2 steps. It donates the first electron in one first step and then donate the second electron here. It fight because it is a reducing agent, as we will see in the future, inshallah. In the second and third year, it fights the free radical, the ROS, reactive oxygen species, if you heard about. The free radicals. Free radicals are very dangerous. They are very dangerous. They cause different diseases. They can cause also cancer. They can cause cell death. They can cause different types of diseases. This is why free radicals are highly dangerous in our body. Vitamin C fight them. Also, the most important thing for you to know about vitamin C role in free radical is regenerate the vitamin E. Now, which one is the more important as for fighting a free radical? Is it vitamin E or vitamin C? Vitamin E is more important than vitamin C, or more efficient as a free radical fighter. Why? Because vitamin E is a lipid soluble. This is why it can penetrate in the cell, to the inside the cell, out of the cell, while vitamin C is water soluble. So its movement in the cell is not easy because it's water soluble. So which one is more efficient in fighting the free radical? Is vitamin E. Why? Because it is lipid soluble. Why vitamin C is very important? Because it regenerates the oxidized form of vitamin E. Vitamin E, once it fights the free radical, it will be oxidized. Then comes the vitamin C. It will reduce the oxidized form of the vitamin E and regenerate it again to fight the free radicals. And lastly, a deficiency of ascorbic acid will cause scurvy, اللي هو مرض الاسقربوط. مرض الاسقربوط. So deficiency of ascorbic acid result in scurvy, a disease that causes sore, spongy gums, loose teeth, fragile blood vessels, and swollen joints, and may cause also anemia. All of these symptoms can be explained by deficiency of hydroxylation of collagen, which is done by hydroxylation of collagen, hydroxylation of proline residue that result in defective connective tissue. شكرا لكم جميعا، بتمنى لكم التوفيق. هذه كانت محاضرة اليوم، فيتامين B9 B12 and C. إذا عندكم أي أسئلة أنا إن شاء الله مستعد لإجابتها الآن. إذا عندكم أي أسئلة بهذه المحاضرة أو بالمحاضرات السابقة تفضلوا.
دكتور
تفضل.
المركبين اللي بيأثروا على تحويل الدمب إلى DUMP.
ايش اسمك؟
أنا مراد الطرامني.
أهلا مراد. أيوه.
كنت أقول هسه هذول المركبين بيمنعوا تحويل DUMP إلى DT.
لا خليك معي بس. وين هاي خليك معي مراد.
أوكي.
بس خلينا نطلع والله هاي شو اسمه البوينتر هذا والله ما نعرف وين هو. مراد خلينا بدون البوينتر. شايفين انتم الماوس هنا؟ شايفه؟ اوكي. شوف مراد، اللي هون عندنا الفوليت اللي هو FH4. الاكتف فورم شو بنسميه؟ FH4. FH4 بيحمل 1 كربون جروب in the form of methylene، فلذلك بيعمل ميثايليشن للدم، فبيحولها ل DTMB، لأنه اليوراسيل موجود بالRNA بينما الثايمين موجود في الDNA فقط. فلذلك ال FH4 بيعمل ميثايليشن للدم وبيحولها ل DTMB. هذا هو النورمال بروسيس. معي مراد؟
معك.
الاف اتش 4 مش بيعمل دونيشن للميثايلين فبتحول لاف اتش 2. بترد الخليه تحول الاف اتش 2 لاف اتش 4 لانه سايكل. نفس الانزيم دايهيدروفوليت ريدكتيز باستخدام ناد بي اتش بعطي الاف اتش 2 ذرتين هيدروجين فبحولها لاف اتش 4. برد الاف اتش 4 ياخذ 1 كربون جروب فروم سيرين فبتحول الى اف اتش 4 وبحمل ال1 جروب على الميثايل جروب ان ذا فورم ال1 كربون على شكل ميثايل جروب على N5 وN10. فالسيرين هون كمان بتتحول لجلايسين. مراد وشو بدك هون؟
السؤال اللي هو كان انه قلت انه هذول المركبين بيمنعوا تحويله مفيد ضد السرطان.
نعم.
انه يمنع تكاثره.
في عندنا ادويه الكيموثيرابي. الكيموثيرابي اللي هي الكيماويات الكيميائي العلاج الكيميائي. حكينا انه عندنا علاج كيماوي اسمه 5 فلورويوراسيل. 5 فلورويوراسيل هذا كيموثيرابي. كيموثيرابي بيمنع الاف اتش 4 انها تعمل ميثايليشن للدم لا يتحول لدي اي تي ام بي. فلذلك ال5 يوراسيل بيعمل بلوك كونا للاف اتش 4 فلذلك الدم ما بتحول الى الدي اي تي ام بي. الدي اي تي ام بي ثايمين واحنا بنعرف انه الثايمين موجود في الدي ان اي. اذا ما في ثايمين ما في سينثيسيس للدي ان اي. ما في سينثيسيس للدي ان اي يعني ما في تكاثر للخلايا. ما في تكاثر للخلايا اذا السرطان وقفنا لانه السرطان هو عباره عن انقسام متواصل مستمر للخلايا. هذا هو السرطان فقط. uncontrolled cell division مثل ما راح نشوف بالفصل الثاني ان شاء الله في محاضره الكانسر وانا راح اعطيكم اياها ان شاء الله. الكانسر هو uncontrolled cell division. اذا الخليه ما لقت القواعد النيتروجينيه هنا الثايمين والنيتروجين بيسز هون ما راح يتضاعف الدي ان اي. اذا ما ضاعف الدي ان اي الخليه ما راح تنقسم. اذا الخليه ما انقسمت اذا وقفنا السرطان. المركب الثاني اللي هو ميثوتريكسيت. ميثوتريكسيت كمان بيعمل بلوك للدايهيدروفوليت ريدكتيز فلذلك الاف اتش 2 ما بتتحول للاف اتش 4. اذا ما في اف اتش 4 نفس النتيجه. الدمب ما بتحول لدي تي ام بي وهذا هو النتيجه. مراد.
السؤال الاساسي اللي هو كان مش هذا المركبين ايضا بأثروا على الخلايا العاديه؟
كل الكيموثيرابي يا أستاذ. الكيموثيرابي مثل ما راح ناخذ ان شاء الله مراد. الكيموثيرابي لما تعطيه انت للمريض بتعرف بتكون ماشي على شفره. تعرف راس الشفره كيف؟ حد. الكيموثيرابي مراد اذا زدته شويه ممكن تقتل المريض واذا قللته شويه ممكن ما يكون له تأثير قوي على قتل الخلايا السرطانية. فلذلك لازم تعطيه بمقدار بيكون دقيق جدا. فلذلك الكيموثيرابي هو سام للخلايا وقاتل للخلايا 100%. وأكثر الخلايا تأثرا بالكيموثيرابي هي الخلايا اللي بتنقسم بسرعة خلايا الدم الحمراء وإلى آخره. فبتأثر هو مواد سامة. أصلا كل الكيموثيرابي هن عبارة عن يعني مواد سامة. فلذلك انت بتشوف اللي بتعالج بالكيموثيرابي شو اثار الكيموثيرابي عليه. ليش؟ لانك بتعطيه سم. انت سم اذا زدت السم شويه راح تقتل كل الخلايا. ليش الخلايا السرطانيه هي اكثر الخلايا كمان تاثرا فيه؟
انها اسرعها انقساما.
يا عيني عليك يا عيني عليك يا مراد. لانه اكثرها انقساما واسرعها انقساما فلذلك اكثرها تاثرا بكم. نعم مراد.
يعطيك العافيه دكتور. هذا هو كان سؤال.
الله يعطيك العافيه. شباب اذا عندكم اي سؤال تفضلوا. إذا عندكم أي سؤال بهذه المحاضرة أو المحاضرات الماضية ما في أي مشكلة.
دكتور.
تفضلي.
يعطيك العافية.
يا ميت مرحبا.
بدي أسأل في آخر سلايد.
نعم. خلينا نروح عليه بس.
دكتور بده يغادر على دي؟
ما في أي مشكلة. اللي ما عنده أي سؤال يتفضل ما في أي مشكلة إن شاء الله. شكرا لكم لحسن استماعكم. تفضلي.
دكتور هلا المرض اللي جاي آخر نقطة هلا هو بصير من deficiency في فيتامين سي مع الهيدروكسيليشن اوف الكولاجين؟
لا هو هذا الفيتامين سي is very important للهيدروكسيليشن.
تمام.
يعني هذا الفيتامين سي بيعمل هيدروكسيليشن للبرولين، فاذا ما في هيدروكسيليشن للبرولين بصير عندنا في ديفكتيف بالكونكتيف تيشيو، بصير عندنا هاي المشاكل.
تمام، شكرا دكتور.
يعني مرحبا. اذا في اي سؤال عن اي المحاضره تفضلوا. مثل ما حكيت لكم خلينا اعيد لكم اللي حكينا عنه، وكل محاضره بعيد لكم اياه. اللي مطلوب منك السلايدات فقط، كل شيء في السلايدات مطلوب منك، كل شيء في السلايدات مطلوب منك، حتى الرسومات، حتى هذا هو يعني انت المطلوب منك انك تميز مثلا في الرسومات فقط. مش مطلوب منك، مثل ما بتعرف كل اسئلتنا هي مالتيبل تشويس، ولكن كل شيء في السلايدات مطلوب منك. يعني مثل ما حكيت لكم بالمحاضرات الماضية، العهد اللي بينه وبينكم هو السلايدات. هون واضح للجميع؟ ادرس السلايدات مليح، افهمهم واحفظهم، وإن شاء الله ما راح تواجه أي مشكلة إن شاء الله في الأسئلة. وبتمنى لكم التوفيق جميعا. وبعيد مرة ثانية إذا في عندكم أي سؤال تفضلوا. أنا ما عندي أي مشكلة، والمحاضرة الثانية مطولة. خلينا نوقف التسجيل.